Alrighty, so I'm going to talk about mutations, but in order to understand mutations, you have to understand what DNA and codons and RNA and all that wonderful stuff is. So over here on the left, I have drawn a lovely molecule of DNA, and you'll notice that DNA pretty much looks like a twisted ladder, and that I've colored the little rungs of the ladder different colors, and there's a reason for that. So these little rungs that link either side of the ladder together, those are called nitrogenous bases, and there are 4 different ones in DNA, and they hold the structure together. There's adenine, thymine, guanine, and cytosine. I may or may not be spelling that last one correctly, but there are these 4 that go together. And you'll notice that in my picture, I drew blue and purple together, and I drew light green and dark green together, and there's a reason for that. In DNA, adenine always matches up with thymine, and guanine always matches up with cytosine. So we could label the light blue A, the dark blue T, the blue G, and the purple C. They always match together like that. Now, the reason for that is because whenever your DNA needs to be copied, or a part of it needs to be transcribed in order to make a protein, the DNA will unzip, and whatever mechanism is copying the DNA will come in, and it knows exactly what the other side should be because all of these pairs need to match up. So if this one was A, this little copier strand here will know to put a T here. If this one was C, this copy strand will know to put a G here, so on and so forth. So it's a nice little system it has. Now, whenever DNA needs to make a specific protein for something, RNA will be the molecule that comes in and makes a little copy of a section of DNA. Now, the difference between RNA and DNA is RNA is only half of a ladder. It'll look something like this. And RNA does not have any thymine in it. It has something called uracil in it. So with RNA, it's uracil, or just U, and then adenine, guanine, and cytosine. So the base pairing rules are a little bit different. A pairs with U, T still turns into A, and then G and C pair with each other like normal. So have these, and have those memorized. Now, your DNA is like thousands and thousands and thousands of these letters long, so your DNA will organize it into these 3 little things called a codon. So let's say TAC is a section. We would call these 3 letters a codon. Now what happens when your RNA goes into your DNA and copies a section down, it will then take the DNA to a structure in your cell called a ribosome, and the ribosome will turn this little codon and all the other codons that follow it into what we call amino acids. And then when you have a bunch of amino acids together, they make a protein. And proteins in your cells do a whole bunch of different stuff that keep you alive. So that is useful. And you can use this thing called a codon wheel to determine what codons equal which amino acids. All these little things on the end here are amino acids. So for example, TAC would translate into T translates into A translates into U, and C translates into G. So this would be your RNA codon here. And the way we use this wheel to figure out what this RNA codon, which amino acid it would be, is we would go to the middle circle here and we would find A. So A is right there in green. And then we would go to the second circle and we would find U in the A section. So U is right there in dark blue. And then we go to the third one and find G, which is right here. So AUG, the amino acid that codes for is MET, is abbreviated as MET, which is actually also a start codon that tells the RNA, hey, start translating here. So let's practice real quick. If I had the DNA strand, let me make up something real quick. All right, so let's say that's my DNA strand. If I wanted to translate it into regular DNA, if I was just making a copy of the original, G would turn into C, C would turn into G, T would turn into A would turn into T, C would turn into G, and T would turn into A. Now, if I wanted to transcribe this original DNA into RNA, Remember, the base pairing rules are a little bit different. G still turns into C still turns into G, T turns into A turns into U, C turns into G, and T turns into A. And then if I wanted to figure out what the codon CGA stands for, I would go back up to my codon wheel. I would find C, and then G, and then A. And that would be the amino acid ARG. And if I wanted to do UGA, I would go to U, and then G, and then A, which is actually a stop codon that tells the RNA, hey, quit translating here. Because if it never quit translating, then each protein would be like millions and millions of base pairs long, and that wouldn't be useful. So that is how all of that stuff works. So going on to the notes parts. So you need to understand all of that in order to understand mutations. There are two different kinds of mutations because there are two different cells in your body. You have inherited mutations, and you are born with those. You get those from your mom or your dad or a random glitch when you were developing. And then you can acquire your own mutations throughout the course of your life in your regular body cells. So say, for example, I have a skin cell that randomly mutates. Chances are that skin cell is not going to negatively affect me because it is just in that one cell, and my one cell does not affect my whole entire body unless it's cancer. Cancer is an exception. But for the most part, my one skin cell that's mutated isn't going to affect the rest of my body. It's going to live and die, and I will never know the difference. However, if I have a sex cell that is mutated and that sex cell ends up becoming a baby, that baby is going to be born with that mutation because it will be in every single cell of their body. There are different things that cause mutations. You can be born with them, but a very poor diet, smoking, being exposed to different toxins, radioactive materials, even certain illnesses can increase your risk of getting a mutation or passing them down to your offspring. A specific kind of mutation is called non-disjunction. This is when chromosomes do not separate correctly the way they are supposed to, and mitosis or meiosis. So instead of each cell ending up with 23 or 46 the way they're supposed to, one might end up with 26 and a half, and the other one might have like 27 or 24 and a half, or however that maths. And like I said, mutations can be located in different areas. If you have a mutation in a body cell, that is called an autosomal mutation, and those live and die with that one cell, and they usually don't affect you that much unless it is cancer. That is bad, obviously. If you are born with a mutation, that is called a sex-linked mutation, and you will live with that for your entire life because, again, it is in every single cell in your body. However, not all mutations are good or bad. Some of them are dangerous, some of them are good, some of them are meh. Now, this is the reason you need to know all the stuff about the RNA and the DNA and the codons and everything. There are different types of mutations. If you have a bunch of letters and one is deleted, that is called a deletion mutation. If you have one that is switched out for a different letter, that's a substitution. If you have one that's added, that's an addition. And then with the chromosomal, again, the non-disjunction, that's when the chromosomes don't separate properly and you have a chunk of something that is not supposed to be there, or you are missing a chunk of something that is supposed to be there. And like I said, mutations are not always good or bad. For example, with the lactose intolerance thing, humans are supposed to be lactose intolerant after a certain age. However, we have a lot of us have a mutation that allows us to continue to consume dairy products after we grow up. I consider that a good mutation because I love ice cream. However, if I mutate and have cancer or sickle cell anemia or anything like that that negatively impacts my quality of life, that would be a bad mutation. Sometimes mutations do not have any effect on you at all, and I will explain why. So going back to our codon wheel here, you'll notice that some things like valine over here, there are 4 different codons that can code for valine. So if I happen to have a mutation that changes G, U, U to G, U, C, I'm still going to end up with valine. So that protein wouldn't affect me at all because I end up getting the same thing overall.